Welcome to the SWAM NPTEL course on Food Microbiology for Safe and Sustainable Food Systems. In the last class we have seen the food spoilage, and in this class we will be seeing the spoilage of meat, poultry, egg and seafoods. And we will be discussing introduction, sources of contamination and causes, changes take place after slaughter, meat spoilage, poultry spoilage, egg spoilage and seafood spoilage. Meat and its product are highly nutritious food that is widely consumed by people all over the world. Meat can be obtained from various birds like chicken, turkey, ducks, etc., or mammals like pork, mutton, buffalo, sheep, etc. And after slaughtering, carcasses and primary cuts are processed to raw or processed food products. It is a nutritious, protein-rich food that is highly perishable and has a short shelf life. The biological and chemical nature of meat leads to its deterioration from the time of slaughter until consumption. Meat and its products such as ham, sausage, cooked meat, dry meats, smoked meats, vacuum packed meat, minced meat, etc., are all susceptible to microbial spoilage. Now, sources of contamination and causes. Most spoilage can be caused by natural processes such as lipid oxidation or autolytic enzymatic reaction that occurs in the muscle after slaughtering. Several factors are responsible for microbial contamination of meat such as bacterial flora of animal, knives, utensils, hands and clothing of the workers, pre-slaughter handling of livestock and post-slaughter handling of meat, handling during slaughtering, evisceration and processing, temperature controls during slaughtering, processing and distribution, type of packaging used, handling and storage, changes that takes place after slaughter, that respiration ceases which stops ATP synthesis. The lack of ATP causes stiffening of muscle. The reduction of oxidation reduction potential due to lack of oxygen. The loss of vitamins and antioxidants causes the development of rancidity. Glycolysis begins, in which most glycogen is converted to lactic acid that reduces pH. The ending of a reticuloendothelial system which leads to the susceptibility of meat to microorganisms. Nervous and hormonal regulation ceases, thereby causing the temperature of the animal to fall and fat solidify. This is the pictorial representation what happens during the pre-rigor mortis. The vital functions stops, deprivation of oxygen and residual oxygen into hemoglobin and myoglobin, and the presence of ATP and phosphocreatine, the aerobic metabolism takes place and which leads to actomyosis and myosin dissociation. During the rigor mortis period, which starts the muscle of meat, during this period ATP and phosphocreatine reservoir exhausting, and due to depletion of oxygen the red meat is converted to brown meat. Anaerobic metabolism starts and no actomyosin and myosin dissociation takes place and highest toughness is resulted. During the tenderization, which is the third phase, toughness decreases, activation of proteolytic enzymes starts, which is composed of calpains, cathepsins, caspases, proteasomes, etc., which introducing the proteolytic enzymes externally. Now the meat spoilage. Fresh meat is subjected to spoilage by its enzymes and microbial action. The autolysis changes cause proteolytic action on muscle and convective tissues and connective tissues and hydrolysis of fats. The survival and growth of microorganisms are influenced by the composition of the atmosphere surrounding the meat. Fresh meat contains nutrients such as sugars, amino acids, vitamins, cofactors, etc., and it had pH of 5.5 to 5.9 and water activity 0.85 that influence the growth of microorganisms. The most common bacteria isolated from fresh meat are bacteria of the genera Acinetobacter, Pseudomonas, Brocothrix, Thermosfata, Flavobacterium, Psychobacter, Moraxella, Staphylococci, Micrococci, lactic acid bacteria, and various genera of Enterobacteriaceae. The microbial pathogens found in fresh meat are Salmonella, Campylobacter, E. coli, Listeria mycocytogenes. Invasion of tissue by microorganisms. Invasion of tissue by contaminating microorganisms takes place after death of animals, and factors affecting the invasion includes microbial load of the gut of slaughtered animal, health and physical condition of the animal before slaughter, method used for slaughtering and bleeding, rate of cooling. Now factors affecting growth of microorganisms. The kind and amount of contamination with microorganism and the spread of these microorganisms in the meat. The physical properties of the meat, chemical properties of the meat, availability of oxygen and temperature. There are two types of spoilage of meat: the spoilage under anaerobic condition and spoilage under anaerobic condition. Now the spoilage under aerobic condition. Under aerobic conditions, bacteria may cause the following: the surface slime, which may be caused by species of Pseudomonas, Acinetobacter, Moraxella, alkali genes, Streptococcus, Leuconostoc, Bacillus and Micrococcus, etc. It is an indication of spoilage often observed before expiry date. Changes in color of meat pigment. The red color of meat called bloom may be changed to shades of green, brown, grey as result of the production of oxidizing compounds, for example hydrogen peroxide, hydrogen sulfide and lactobacillus and leuconostoc are basically responsible organisms for this. Changes in fats. The oxidation of unsaturated fats may take place chemically in air and may be catalyzed by light and copper, for example oxidative density. The causative organisms are Pseudomonas and Achromobacter are responsible for oxidative density or by certain yeasts. Now, the surface color discoloration due to pigment forming bacteria. A red spot may be caused by Serratia marcescens or other bacteria with red pigment. Pseudomonas cincinnati can impart blue color to the surface. Micrococcus or Flavobacterium imparts yellow color. Now, off colors and off taste. Taints are undesirable odors and off taste that appear in meat. Souring that gives the sour odor that may be due to volatile acids, for example formic, butyric, propionic acids by the action of microbes. Sometimes cold storage flavor or taint observed in meat and it's nothing but stale flavor. In case of musty or herby flavor, meat contaminated with actinomycetes are the causative agent for the same. Now, aerobic growth of molds may cause stickiness, that is the surface becomes sticky. Whiskers, when meat is stored in frozen condition, mycelial growth may take place without sporulation. Black spot is caused by Cladosporium herbarium. White spot caused by Sporotrichum carnis. Green patches are caused by Penicillium such as Penicillium expansum, Penicillium oxalicum. Decomposition of fats is caused by many molds produce lipases and cause lipolysis of fats. This is the pictorial representation of gas production which is caused by Pseudomonas species, Shewanella species and Viscella species, whereas the slime is produced by Vicinella viridensis, Vicinella viridescens, Lactococcus mesenteroides and Pseudomonas species, whereas discoloration of meat is caused by Corynebacterium, Pseudomonas and Serratia species, whereas the luminescence is produced by Photobacterium, Hafnia species and Vibrio quingensis. Vibrio quingensis. Spoilers under anaerobic condition. Facultative and anaerobic bacteria are able to grow within the meat under anaerobic conditions and cause spoilage. Following changes occurs in such conditions. Souring. It imparts sour taste to meat due to acids such as formic, propionic, acetic, etc. Meat's own enzymes are responsible for it. Putrefaction. True putrefaction is the anaerobic decomposition of protein with the production of foul smelling compounds such as hydrogen sulfide, indole, ammonia, amines due to Clostridium species. Taints. Taints are undesirable odors and tastes that appear in a meat. Clostridium species have been associated with the production of large amount of gas in vacuum packed beef, accompanied by foul-off odors. Gas production like carbon dioxide by lactic acid bacteria without extensive off odors may be associated with vacuum packed beef and pork. Pasteurized canned products. Bacillus species, Clostridium species, Enterococci, Streptococci and heat resistant lactic acid bacteria. Refrigerated canned products are spoiled by Streptococcus species. Now the poultry spoilage. Poultry meat, like meat of other animals, is also susceptible to contamination by various sources. Contamination of skin and lining of the body cavity takes place during various processing operations. The organisms of great importance in poultry are Salmonella species and Campylobacter jejuni. Several gram-negative psychotropic bacteria, for example, Pseudomonas, Acinetobacter and Flavobacterium has also been isolated from poultry carcasses. Freezing further reduces the number of microorganisms in the poultry meat, provided the temperature is maintained quite low, that is -18 degrees Celsius or below. Sources of poultry contamination. The skin of live birds may contain numbers of bacteria averaging 1500 per centimeter square and could also be derived from the feet, feathers and feces. Contamination of the skin and the lining of the body cavity occurs during washing, plucking and evisceration. The process of sticking and bleeding can also introduce contamination. Knives, clothes, air and hands and clothing of the workers can serve as intermediate sources of contamination. After the handling of meat, contamination can come from carts, boxes or other containers. This is the pictorial diagram from which the poultry gets its contamination, like from the poultry house environment, that is the exterior, and it may come from the soil and grass, personnel and vehicles, wild and farm animals, feed mill, water fowls. Or it may come from the interior, that is the rodents, pests, darkling beetles or flies, fans, blades and floors. Or it may come from the hatcheries, which will go to the chicks and feed and water consuming, feed and water consumed by the chicks and the feces and litters, and then it will go, this process is continued to contaminate the poultry meat. Now egg spoilage. Eggs are a highly nutritious food that contains proteins, minerals, fats, iron, phosphorus, vitamins like A, B, D, E and K. The eggs most commonly consumed by humans are the eggs of hens, ducks and quails. The chicken's egg in particular is very popular, consists of the yolk 30 to 33%, albumin 60% and the shell 9 to 12%. Egg yolk is about 50% of protein, of which the main ones are lipoproteins, lipovitellins and lipovitellinin. Egg albumin is about 12% protein, of which the ova albumin is 54%, corn albumin is 13%, avidin 0.05%, ova transferrin 10%, ovomucoid 11%, ovomucin 1.5 to 3% and lysozyme 3.5%. The shell of the egg is a rigid structure made largely of calcium carbonate on an inorganic matrix. Internally, there are two shell membranes and the inner one that acts as a barrier when an organism penetrates the cell. The hen's egg is an excellent example of a product that normally is well protected by its extrinsic and intrinsic parameters. This is the pictorial diagram of different egg composition. This is the shell, the outermost is the cell, the outer covering of egg composed of largely of calcium carbonate. It may be white or brown depending on breed of chicken. The color does not affect egg quality, cooking characteristics, nutritive value or shell thickness. The another part is air cell. The pocket of air cell formed at large end of the egg. This is the large end of the egg, caused by contraction of the contents during cooling after laying. Increases in size as eggs ages. The another part is shell membrane. The two shell membranes are there, that is inner and outer shell membranes surround the albumin, provide protective barrier against bacterial penetration. Air cell forms between these two cell membranes. The thin white albumin, it is the nearest to the cell, spreads around thick white of high quality egg. The thick white albumin, the major source of egg riboflavin and protein. It stands higher and spreads less in higher grade eggs, thins and becomes indistinguishable from thin white in lower grade eggs. Another is chalazi. Chalazi is a twisted, this is the chalazi, it is a twisted cord-like strands of egg white. It anchor yolk in center of the egg. It helps yolk to be in the center of the egg. Prominent chalazi indicate freshness. Another is vitelline, that is the yolk membrane. This holds egg yolk contents. Now the yolk, this is the yellow portion of egg. Color varies with feed of the hen but does not indicate nutritive content. Major source of egg vitamins, minerals and fat. Now the germinal disc, this is the germinal disc from where the chickens are formed. Now, extrinsic or external protection. A fresh egg has three structures which is effective to some extent in retarding the entry of microorganisms. The outer waxy shell membrane, the shell, and the inner shell membrane. The intrinsic or internal protection. Lysozyme present in egg white. This enzyme is quite effective against gram positive bacteria. Avidin present in egg white, which forms complex with biotin, thereby making this vitamin unavailable to microorganisms. This is the egg component that we have already discussed, that this is the yolk, this is albumin, and this is the hard shell. This hard shell is composed of 5 parts like external layer, then prismatic layer, then mammillar cone layer, then shell unit, organic shell, membrane. And these are the barriers which protect the egg against the waste handling and environmental conditions, microbes and forms, etc., that contaminated acquired by that. The egg white has got pH of 9.3 and contains cone albumin, which forms a complex with iron, thus rendering it unavailable to microorganisms. In addition to antimicrobial substances, hence egg albumin contain ovotransferrin, which chelates or binds metals, for example iron, ovoflavoprotein, which binds riboflavin. At its normal pH, that is 9 to 10, egg albumin is cidal to gram positive bacteria and yeast at both 30 degree and 39.5 degree Celsius temperature. In addition of iron reduces the antimicrobial properties of egg albumin. On the other hand, the nutrient content of the yolk material and its pH 6.8 makes it an excellent source of growth for most microorganisms. Contamination sources and causes. The eggs when laid are usually sterile, therefore the contamination occurs after it is laid due to various reasons, like by the fecal matters of the chicken, by specks of dirt or materials used for the cage or nest, water used during washing of eggs, equipment used during processing and handling, materials used to pack the eggs, the temperature used during storage, presence of eggshell cracks or micro cracks. This is the pictorial diagram of egg spoilage, and these are the causes. The causes includes the mechanical contamination due to fecal matters of egg and handling, the microbial spoilage, that is due to bacteria and fungi, and micro spoilage. Non-microbial spoilage includes loss of moisture and loss of egg white during long term storage, changes in physical state of egg contents like at a storage temperature and relative humidity, and the preservation techniques of egg spoilage includes the direct methods and indirect method. The direct method is further divided into two, that is the thermal method and non-thermal method. Thermal method includes the hot water treatment, steam, microwave, radio frequency heating, freeze drying or cryodesiccation, radiation, infrared radiation, thermo-ultrasonification, whereas the non-thermal techniques includes using organic substances, using chemical substances, mechanical washing, using neutral electrolyzed water, physical, biological, ultrasonic, atmospheric steam, pulse light, cold atmospheric pressure plasma, UV radiation, ozone and biological fumigation. The indirect factors that includes the age and genotype of the bird, nutrition, heat stress, health status, housing systems and storage conditions. Causes of egg spoilage during storage. Upon storage, thick white loses water to the yolk, resulting in a thinning of yolk and shrinkage of the thick white, which resulted in the direct contact of yolk and inner membrane, where it may be contaminated directly by microorganisms. Once the yolk is contaminated, bacteria grow faster because of high nutrient content and produce H2S, a byproduct of protein and amino acid metabolism, which is foul smelling compound. Yolk becomes runny and discolored. Molds generally multiply first in air sac where oxygen favored the growth. Under the high humidity conditions, molds may be seen growing over the outer surface of eggs. Under low humidity and temperatures, surface growth is not favored, but egg lose water at faster rate and other undesirable changes occur. The microflora of the egg shell is dominated by gram-positive bacteria such as Staphylococcus, Streptococcus, Aerococcus, Bacillus and Micrococcus, and gram-negative bacteria such as Salmonella, Escherichia and Alkaligenes species. The bacteria such as Pseudomonas, Proteus, Alkaligenes, Enterobacter, Serratia, Stenotrophomonas, Cloaca, Acinetobacter, Moraxella and Citrobacter species are responsible for making egg rotten with off odors and color change. Yellow pigmentation of the shell membrane occurs in eggs due to the action of Flavobacterium or Cytophaga species. The molds that are responsible for causing egg spoilages, including species such as Penicillium, Cladosporium, Alternaria, etc. The early mold growth in egg surface is termed pin spot molding, and the final stage of spoilage is termed fungal rotting. This is how to test whether egg is good or bad. When it is placed in a in a bowl containing water, the fresh egg settles down in the bottom. A week old egg is stand in inclined position, whereas the stale that is stands in the bowl in vertical position, whereas the very old they will on the surface, they float on the surface. Now the seafood spoilage. Fish is a very perishable, high protein food that typically contains a high level of free amino acids. Lipid content of the fish is up to 1 to 20%. Fish flesh generally contains 15 to 20% protein and less than 1% carbohydrate. Non-fatty fish like teleost, haddock, fat levels are only about 0.5%, but the fatty fish like mackerel and herring levels can vary between 3 to 25%. Microbes metabolize amino acids producing ammonia, biogenic amines like putrescine, histamine and cadaverine, organic acids, ketones and sulfur compounds. Degradation of lipids in fatty fish produces rancid odors. Marine fish and some freshwater fish contain trimethylamine oxide that is degraded by several spoilage bacteria to trimethylamine, which is responsible for fishy off odors. Spoilage in fish generally accompanied by change in its physical characteristics. Changes in color, odor, texture, color of eyes, color of gills and softness of the muscle are some of the characteristics observed in spoiled fish. Spoilage is caused by the action of enzymes, bacteria and chemicals present in the fish. Now, types of spoilage in fish. The oxidative spoilage. Lipid oxidation is a major cause of deterioration and spoilage of fish that contain high oil or fat content stored in their flesh. Oxidation typically involves the reaction of oxygen with the double bonds of fatty acids. In fish, lipid oxidation can occur enzymatically or non-enzymatically. Lipid oxidation promotes protein denaturation, modification of the protein, electrophoretic profiles, nutritional losses and endogenous antioxidant systems losses. Lipid hydrolysis and oxidation causes belly burst in fish, in which the enzymes and microorganisms of the digestive tract cause massive gas development. Now, the enzymatic spoilage. After capture, biological and chemical changes take place in dead fish due to the action of various enzymes found in fish. The digestive enzymes cause extensive autolysis, which results in fish muscle softening, rupture of the belly wall and drain out of the blood. Several proteolytic enzymes are found in fish that contribute to degradation of fish muscle and fish products during storage and processing. Proteolysis is responsible for the degradation of proteins, which leads to fish spoilage by microbial growth. Now, the microbial growth. The fish flesh is composed of proteins, fats, carbohydrates, water and amino acid compounds such as trimethylamine oxide, urea, taurine, creatine, free amino acids and traces of glucose, etc. The internal tissues of fish is generally considered sterile. Bacteria are present on the slime layer of the skin, gill surfaces and the intestine. The microbial growth in fish is the main cause of fish spoilage and produces amines, biogenic amines, organic acids, alcohols, aldehydes and ketones with unpleasant of flavor. The high water activity and low acidity, like pH is more than 6 of the fish, result in the fast growth of microorganisms that leads to undesirable changes in appearance, texture, flavor and color, reducing its quality. At room temperature, Bacillus, Clostridium, Escherichia, Micrococcus, Proteus, Sarcina and Serratia may predominate. For unpreserved fish, spoilage is caused by gram-negative fermentative bacteria such as Vibrio-necia, whereas psychrotolerant gram-negative bacteria such as Pseudomonas species and Salmonella species tend to spoil chilled fish. The fish spoilage is also caused by psychotropic aerobic or facultative anaerobic gram-negative bacteria such as Pseudomonas, Moraxella, Acinetobacter, Salmonella, Salmonella putrescens, Vibrio, Flavobacterium, Photobacterium and Aeromonas. The gram-positive bacteria such as Staphylococcus species, Micrococcus, Bacillus, Clostridium, Corynebacterium, Brocothrix, Thermophaga and Streptococcus are found in fish. The lactic acid bacteria can predominate in fish storage under vacuum or carbon dioxide storage. Some parasites can also be transmitted by fish, including tapeworm, for example, Diphyllobothrium latum, nematodes like Anisakis simplex and Capillaria philippensis, and trematodes like Opithorchis and Paragonimus. Spoilage compounds are produced by microorganisms during the storage of fresh fish.